Good afternoon and welcome to the Alliance for Eye and Vision Research's Congressional Briefing on Dry Eye Disease, conducted during Dry Eye Awareness Month. I'm Dan Ignazewski and I am the Executive Director for the Alliance for Eye and Vision Research, or AEVR. And AEVR is hosting this briefing in conjunction with the Tear, Film, and Ocular Surface Society, or TIFOS. Before we begin, I just want to take care of some quick housekeeping. You'll notice that during this Zoom briefing that only speakers will be visible or heard throughout the briefing. If you have questions about dry eye, please use the Q&A box at the bottom of your screen. You can also use the chat box for support or other communications, but please submit your questions throughout the presentations and we'll try to answer all the questions in our follow-up. I also want to confirm that this briefing is being recorded and will be available on the AEVR website and YouTube channel. And will also be shared on the Association for Research in Vision and Ophthalmology website and YouTube channel as well. Now that I've covered some of the logistics, let's move on to the program. AEVR's mission is to ensure the best eye and vision care for all Americans through the education of congressional legislators, government policymakers, coalition partners, the media, and consumers about the value of eye and vision research, and we welcome you all here today. AEVR also serves as the privately funded Friends of the National Eye Institute, or NEI, and we launched our Research Saving Sight Restoring Vision initiative last year. Today's briefing is AEVR's third Research Saving Sight Restoring Vision event of 2022. The Research Saving Sight Restoring Vision initiative is a sustained activity to educate about eye and vision conditions and highlight the enormous strides that the National Eye Institute funded research continues to make in terms of new approaches to diagnostics, interventions, and therapies for eye and vision care. It also highlights the tools that support these new approaches, such as imaging, big data, and artificial intelligence. As I said, July is Dry Eye Awareness Month, and we've worked with partners across the eye and vision community to recognize the importance of addressing dry eye disease. I want to take a moment now to introduce Amy Sullivan, who is the Executive Director for TIFOS and has collaborated on this briefing so that she can share a little bit about dry eye disease and TIFOS and the importance of addressing dry eye disease. Amy?
Dry eye disease is a global epidemic, and increased funding for dry eye research is paramount. Thanks, Dan, for having us here today. Hi. I'm Amy Sullivan, Executive Director of the Tear, Film, and Ocular Surface Society. I'm here today in collaboration with AEVR and the vision community, representing Dry Eye Awareness Month. TIFOS has a mission to advance the research, literacy, and educational aspects of the scientific field of the tear, film, and ocular surface. Together, we are creative, passionate, inspirational, enriching, international, collaborative, committed, and inquisitive. We collaborate with eye care professionals in more than 100 countries. We've given presentations and shared information with hundreds of thousands of eye care specialists and patients around the world. Since the incorporation of TIFOS in 2000, dry eye research has actually more than doubled. Dry eye disease is a global epidemic, and dry eye disease is a leading reason for visits to the eye doctor. Dry eye is a more than $60 billion burden on US society. Dry eye research can improve patients' quality of life. Dry eye research can reduce healthcare costs. The US is a world leader in vision research and scientific training. Dry eye disease research will help individuals return to productive, independent, and quality lives. Dry eye disease research funding improves the development of the next generation of vision scientists. Together, we make things happen. And today you will hear presentations from Dr. Maria Marculi, Dr. Anat Galor, and Dr. Tannen Schmidt. Thank you again for joining us today to recognize July as Dry Eye Awareness Month. And thank you to all of our sponsors who make this education possible. And thank you to Congress, who we hope will increase the funding for dry eye research. Thank you again.
Thank you, Amy. We've enjoyed working with TIFOS and bring you all this briefing. I also want to recognize some others who have helped make today's briefing possible. Today's briefing is supported by several major vision organizations and companies, and I'd like to thank each of them for helping to support today's briefing. That includes the Research to Prevent Blindness, TIFOS, and the Association for Research in Vision and Ophthalmology. I also want to thank our industry partners, Genentech and Novartis, for providing event support to make today's briefing possible. Before we introduce our speakers, I want to share a little more about dry eye disease. Dry eye disease is when an individual's nerves and tear film don't properly keep your eyes wet, and this can grow into be a debilitating condition for many. With our lifestyle impacting our eyes more and more, we expect to continue to see the incidence of dry eye disease grow in the United States and throughout the world. Many of you may be familiar with some of the current treatments to manage dry eye disease and may have even tried some of the over-the-counter solutions yourself when your eyes feel dry and strained. But there are also prescription solutions, lifestyle considerations, tear duct plugs, and surgery that can help manage dry eye. As Amy mentioned, dry eye is costly. The annual direct healthcare costs are substantial, and the indirect societal costs are significantly higher. Each year, AEVR's Dry Eye Disease Briefing features internationally recognized experts in dry eye disease, and today is no different. First, we'll hear from Maria Marcouli, who is an associate professor at the School of Optometry and Vision Science at the University of New South Wales. Her research interests relate to ocular surface and health and disease, and she is a dual Ezel Fellow from the American Optometric Foundation and was on the TIFOS subcommittee of the Contract Lens Discomfort Workshop and on the Pathophysiology subcommittee of the TIFOS Dry Eye Workshop too. She's the deputy editor of the Clinical and Experimental Optometry and a postgraduate coordinator for the School of Optometry and Vision Science at UNSW. She's also the Australian TIFOS ambassador and is chairing the nutrition subcommittee of the upcoming TIFOS workshop on a lifestyle epidemic, ocular surface disease. Maria?
Hi, I have a question for you. Is fast food a fast track to dry eye disease? Before we attempt to answer that question, allow me to share with you why I think the answer to that question matters. As you can probably tell, I am Australian. I was born and bred in the harbour city of Sydney, where I work as an optometrist and as an academic at the University of New South Wales. That means that I get to do all the things I enjoy. I get to help people through my clinical work, teach undergraduate and postgraduate students, and conduct research into ways that we can continue to help people into the future. One of the conditions that I most commonly encounter in our clinic is dry eye disease. For the patient, this can mean anything from a slight discomfort, including a sandy, gritty sensation, stinging or burning, right through to extreme discomfort to the extent that they cannot be in an air-conditioned room, they cannot go outside when it is even slightly windy, and they cannot sit in a well-lit office or be in the sun, as any of these conditions will mean that they cannot open their eyes from the excruciating discomfort and the reflex tearing. Things that many of us take for granted can often be debilitating for the patient with dry eye disease. Their vision is often also affected, with things being blurry in between blinks or there being halos around lights, especially when driving at night. When they look at themselves in the mirror each day, they see these red, angry-looking eyes, and they are often asked by well-meaning friends, family, and colleagues if they are okay. So it goes without saying that their quality of life is significantly affected. Ideally, what we want is to have a healthy 3-layered tear film and healthy glands which produce each of these 3 layers. The yellow layer is the lipid or oily layer, which is produced by the glands in our eyelids, known as the meibomian glands. This layer sits on the aqueous layer, shown here by the blue, and this is produced by the lacrimal glands. And the bottommost layer is the mucus layer, which is produced by the cells of our cornea and conjunctiva. So oil sits on water to prevent evaporation of the tear film. So simple, yet so clever. If we have a deficiency in any one of these layers or damage to any of the structures which produce them, dry eye disease can result. When we evaluate patients with dry eye, we often see that the openings of the glands which produce the oily layer are blocked. And when we invert their eyelids and observe them in infrared light, we can see that compared to this healthy eyelid, a large number of those oil-producing glands are starting to die off. And when we measure the thickness of the lipid layer produced by these glands, we can see that compared to a healthy eye, which has a thick layer of oil visible, the eye with dry eye disease does not have a perceptible oily layer, and the patient here really struggles to keep their eyes open. When we evaluate the aqueous layer, we will often see that the lacrimal gland is not producing enough volume, and all of this results in a tear film that is not stable. In the photo on the right, we have used a fluorescein dye to stain the tear film, and in using a blue light with a filter, we can see black areas indicating areas where the tear film has thinned within a few short seconds, resulting in all the symptoms that we have discussed. Perhaps the worst of all is when we see that these patients are at the point of no return, with ocular surface damage, specifically to the conjunctiva, the cornea, and the eyelids, due to this loss of lubrication between these tissues. In addition, the eyes and eyelids are red and inflamed, and we know that the eye is inflamed because when we collect and analyze the tears for inflammatory products, we find that these are upregulated in dry eye disease. So we have a person here who is uncomfortable, has trouble getting through their day, their eyes look perpetually red, and when we look at them with our equipment, we can see that they are at risk of permanent damage and being at that point of no return. A lot of what we do in the clinic room is to provide these patients with palliative relief. We ask them to buy artificial tears and to instill these into their eyes multiple times a day, often for years at a stretch, at great expense to the patients and often at great inconvenience. We may also block the drainage duct, either temporarily or permanently, and ask them to wear these very attractive looking moisture chamber spectacles to prevent evaporation of their tear film. Patients often will need to take anti-inflammatory eye drops in order to reduce the perpetual inflammation, and these eye drops themselves place patients at increased risk of developing severe infections and even glaucoma into the future. We can also conduct painful procedures in the clinic room to express the contents of the glands in order to express as much lipid into the eye. In the video on the left, we can see that on expression of the eyelids, the glands are expressing this healthy looking oil substance, which is what we want. On the right, we can see that the glands are expressing almost a toothpaste-like expression, and this is less than ideal for the maintenance of a healthy tear film. Remember, we need oil on water, not butter on water. We can also apply various instruments to encourage their eyelids to expel their contents in the hope that we can reactivate the glands that produce the various parts of the tear film. There is no cure, we are merely managing the condition. Based on the data from the National Health and Wellness Survey, 6.8% of American adults, approximately 16.4 million people, have been diagnosed with dry eye. The average annual cost to a patient is approximately $783 US. The overall burden of dry eye for the US healthcare system is estimated at $3.84 billion. From a societal perspective, the average cost of managing dry eye is estimated to be a lot more at $11,000 per patient and $55.4 billion to the US society overall. This condition poses a substantial economic burden on the patients and on society. So what if I told you that there might be something that we can do to reverse or even prevent the development of dry eye disease in the first place and potentially reduce the burden for future generations? We're told continually that we are what we eat, but that might mean more than just for our baseline or our blood cholesterol levels. Potentially the most studied dietary components relating to dry eye disease is omega-3 and omega-6 polyunsaturated fatty acids. With numerous randomized controlled trials and systematic reviews and meta-analyses. Both omega-3 and omega-6 fatty acids must be consumed in the diet, as the human body is incapable of synthesizing them. Omega-3 fatty acids are present in fish such as salmon, mackerel, tuna, herring, and sardines, in chia seeds, walnuts, and in plant oils such as flaxseed, soybean, canola oils, fish oils, and krill oils, as well as dietary supplements. Omega-6 fatty acids can be found in healthy foods such as tofu, walnuts, and peanut butter, but they're also present in processed snacks, fast foods, cakes, fatty meats, and cured meats. Consumption of omega-6 fats has dramatically increased over recent years, And this is important because omega-6 and omega-3 share the same enzyme system, creating a competitive environment between them that is either primarily pro-inflammatory in diets that are dominated by omega-6 or anti-inflammatory in diets dominated by omega-3. The ratio of omega-6 to omega-3 matters when it comes to our health, with the ideal ratio being 4:1. An imbalance in this may have important implications for the development of chronic diseases, such as cardiovascular disease and cancer. In dry eye disease, researchers hypothesized that omega-3s might reduce the risk of dry eye and relieve its symptoms because of their anti-inflammatory effects. In the women's health study of more than 32,000 women older than 45 years shown in this graph here, a higher omega-6 to 3 ratio was associated with a significantly increased risk of dry eye disease, with those consuming higher levels of omega-6 having a 2.5 times higher risk of developing dry eye than those who consumed the recommended ratio of 4:1. Additionally, a 30% reduction in dry eye risk was found with each additional gram of omega-3s consumed each day. There's a lot that we don't know though, such as what the optimal dietary intake needs to be to prevent and manage dry eye disease. When it comes to omega-3 supplements, results from clinical trials have had mixed results, often due to differences in the study design. The graph shown here is from a meta-analysis that investigated omega-3 versus placebo and included 17 randomized controlled trials that had over 3,000 patients. The authors reported that omega-3 supplementation decreased dry eye symptoms as well as the clinical signs of dry eye disease. We need more research to more clearly define who goes into these studies, to use a set core outcome measures, and to determine the optimal dose, composition, and duration of supplements that are necessary. Another area receiving some attention with regards to dry eye is the Mediterranean diet. The principal aspects of this diet include proportionally high consumption of olive oil, legumes, unrefined cereals, fruits and vegetables, moderate to high consumption of fish, dairy products and wine, and low consumption of red and processed meat and sugar. The Mediterranean diet has been reported to be useful in chronic inflammatory diseases due to its anti-inflammatory properties. Given these properties, it might be expected to also have benefits in dry eye disease. To that end, a higher adherence to the Mediterranean diet has been associated with a lower likelihood of developing a type of dry eye disease known as Sjogren syndrome. Another study randomized participants with this condition to one of two groups, either a Mediterranean diet supplemented with extra virgin olive oil and nuts, or a hypercaloric Mediterranean diet, as well as physical activity. In both groups, both the signs and symptoms of dry eye disease improved after 6 months, although more so in the group with exercise. You'll notice that one common thread throughout this talk has been the role that inflammation plays in dry eye. The corollary here being that if we manage inflammation, then we can also manage dry eye disease. Your gut microbiome is made up of trillions of microorganisms and their genetic material that live in your intestinal tract. As well as influencing gut motility, the gut microbiome also plays an important role in the regulation of low-grade chronic inflammation in different parts of the body, including the eye. Ecological shifts within the gut microbiome can induce imbalance or dysbiosis, which is associated with chronic disease. Dysbiosis can be induced by our modern lifestyle, including dietary changes, antibiotics, artificial sweeteners, vitamin deficiency, smoking, and saturated fats. In dry eye disease, severe gut dysbiosis has been found to be significantly more prevalent in patients with Sjogren syndrome, that type of dry eye disease I mentioned earlier, compared to healthy controls, suggesting that the severity of gut dysbiosis is associated with the severity of clinical manifestations in dry eye. Also, reduced gut microbiome diversity in dry eye patients compared to healthy individuals has also been reported. So an obvious path to follow here is to explore whether modulating the gut microbiome plays a role in treating dry eye. And there are 3 common methods for altering the gut microbiome. One is fecal microbiota transplantation, and the other is application of pre- and probiotics. Prebiotics boost the function of probiotics and other good bacteria while probiotics include beneficial bacteria and can be obtained from fermented foods or commercial supplements. In this study, a combination of pre and probiotics was given to a group of people with dry eye for 28 to 30 days, shown here in the black bars. When compared to the control group, the prebiotic group was found to have an improvement in both the signs and symptoms of dry eye disease. Now, the field of modulating the gut microbiome as an intervention to treat dry eye is relatively in its infancy. And while these results are encouraging, we need a lot more comprehensive investigations in order to inform both patients and clinicians about the practical applications of such treatments, such as the proper dosage, duration, and formulation of these supplements. Other aspects of our diet can also potentially impact on our eye health and our risk of developing dry eye. Sugar, for example, is associated with inducing metabolic syndrome and development of glucose intolerance and weight gain. This can lead to systemic inflammation, although there is no research yet on the direct impact on dry eye. The major vitamins playing a role in ocular surface diseases include vitamins A, B12, C, and D, and their deficiency is well known to cause dry eye disease. Food additives added to food to improve freshness, flavor, texture, or their appearance are largely unexplored in dry eye. And contaminants as a result of food production, such as PCBs, can leach. And while some of these are known to have bioactivity in the human body, their role in dry eye is not known. Bisphenol A, a chemical compound used in the production of plastics, including food and drink containers, has been found to increase inflammation in the eye. So we clearly need to understand a lot more about all of these factors and their role in dry eye disease. This brings me back to the original question I posed. Is fast food a fast track to dry eye disease? Fast foods tend to be low in whole grains, fruit, vegetables, nuts, and seeds, but high in sugar, sodium, and processed meats, which along with high intakes of alcohol are leading factors in early death and disability. Much of the research to date relating to nutrition has really revolved around conditions such as cardiovascular disease, cancer, and diabetes. The data we have so far seems to indicate that a diet high in fast foods will contribute to dry eye. We need to understand a lot more, such as which factors in our diet are helpful in managing and preventing dry eye disease, what the recommended daily intake should be in the case of managing and in the case of preventing dry eye, which supplements are beneficial and what their dosages should be, and which factors are detrimental. By understanding the impact of nutrition on the eye and specifically dry eye disease, we can reduce the burden of dry eye, not only on ourselves, but also on future generations. I'd like to acknowledge my colleagues involved in collating this information as part of the ongoing TFOS Lifestyle Epidemic Nutrition Workshop, and I thank you for your time.
Thank you, Maria, for that great introduction to dry eye disease and for helping us understand the importance of nutrition on eye health and how it impacts dry eye. Next, I'd like to introduce Annette Galore, who is a cornea and uveitis trained specialist with a dual appointment at the Miami Veterans Affairs Medical Center and the Bascom Palmer Eye Institute at the University of Miami Miller School of Medicine. Dr. Galore completed an ophthalmology residency at the Cole Eye Cleveland Clinic, a uveitis fellowship at the Wilmer Eye Institute at Johns Hopkins University, and a cornea and external disease fellowship at Bascom Palmer Eye Institute. Dr. Galore currently runs the Ocular Surface program at the Miami VA and has focused her research on understanding mechanisms underlying chronic ocular surface pain, with an emphasis on studying new diagnostic and treatment modalities. Over the years, her work has been funded by the NIH, VA, and DoD, and she has published and lectured widely on these topics. She has participated in several dry eye-related committees, including the TFOS Dry Eye Workshop, and she's actively involved with a number of organizations, including ARVO, AAO, and Ocular Microbiology and Immunology Group.
Hi, my name is Annette Galore. Thank you all for having me here to talk to you a little bit about why dry eye is such a pain. But I actually want to flip and ask the question in a different way. What is dry eye? And the reason I say that is because dry eye is actually not one disease. So when you hear the word dry eye, I want you to stop and think and ask, what kind of dry eye are we talking about? And I like to think of dry eye as an umbrella term. And under that umbrella term are lots of different subtypes. So some people don't have enough of the water component in tears. Others don't have enough of the oil component in tears. Others have abnormal anatomy that causes their tears to be abnormal, but it's actually an anatomic issue. And some people have abnormal nerves. And any of these can show up as what we call dry eye. And what makes it more confusing is you can have any of these subtypes in isolation, or they can appear along with a systemic disease, things like Sjogren's and graft versus host disease. And that's why it's not enough to use the word dry eye. Now, here's another way of looking at it. It's so easy to think about dry eye as a tear problem or as a water problem. But if you look at the composition of tears, you start seeing it's much more complex. It's actually a bilayer. You have your lipid layer here on top, and then a mucoaqueous layer here. And each of these layers is made by a different gland. So the lacrimal gland, they make the water component, that's in blue. And the meibomian glands, they make the lipid layer here on the outside that protects the water from evaporating. And then there's this mucin layer made by goblet cells on the conjunctiva. And so every time you blink, this tear layer has to be formed. And you can imagine it has to be formed from components in different locations. So any bumps or lumps or problem with your eyelids or problems with blinking is going to cause the tear film not to form normally. Now, why is this so important? It's so important because dry eye is common. Some studies have found that 5 to 30% of the population have some form of dry eye. And it's not just that it's common, the presence of dry eye symptoms negatively affects quality of life. So I want you to stop for a second and think, do you feel your eyes? Because you're not supposed to feel your eyes. So imagine that all day you felt your eyes. You can see how that may make reading, computer work, television watching, driving more difficult. And so going back to what underlies dry eye, we talked a little bit about how dry eye could be a tear problem or it could be a nerve problem. And when you hear the word dry eye, it's easy to think, well, it's a problem with tears, but this idea that nerves may be abnormal in dry eye is a newer concept. And it's been one of my research focuses is trying to show that nerve function is not equal across individuals. And that's true individuals with and without dry eye symptoms. And so I have this machine where I can puff air of different intensity and ask people when they feel the air. We call that detection threshold. When do you feel a puff of air? And there are some people that feel it when it's very faint and other people that feel it when it's stronger. And it's a huge spread if you look at the 10th to 90th percentile. And even with that huge spread, about a quarter of my population is outside that spread, some being hypersensitive and some being hyposensitive. And so the punchline is, you and I probably have different thresholds where we would feel that puff of air. And the reason that's important is because we have these ways to measure tear production. We put little pieces of paper in the corner of the eyes and we measure the wetting at 5 minutes. And so this means that at 5 minutes, the tears have wetted down to 4 millimeters. And that's considered aqueous deficiency or not having enough water. The thing is, some people have this low tear production and they come in and say, my eyes feel dry. But others have low tear production and they don't complain at all. And so we now believe that it's the differences in how your corneal nerves interpret what's happening in your environment that explain these differences. And so it's so easy to just blame the Schirmer strip, that little piece of paper that we use to measure tear production, but actually the Schirmer strip is trying to say it wasn't me. So we now understand that it's not enough to just measure tear parameters, but that it's also important to consider nerve function, because your cornea is supposed to be scratch-free, and everywhere you see white, that's a little tiny scratch. And you would imagine that some people who have such scratchy corneas would come in complaining, but they tell me, hey, my eyes feel great. And we now understand that it's probably because their nerves are just not appropriately understanding what's happening in the environment. The nerves are not sensitive enough. And we actually have a word for that. We call it neurotrophic keratitis. And then we have eyes that have absolutely no scratches. They look amazing. And yet they tell me, oh, my eyes feel dry and burning and aching. And now we also have a word for that. We call that a neuropathic phenotype. And so if that's not complex enough, you can have any nerve abnormality and any tear abnormality coexisting. So you can have neurotrophic or neuropathic phenotype, and you can have low tear production, you can have oil problems, you can have anatomic abnormalities. And that is why dry eye really is a little bit of a pain, because it's a complex disorder that can manifest in multiple different ways, and it has lots of underlying potential contributors. So our job is not to give up, not to get discouraged, but to figure out what is contributing to the dry eye symptoms and signs that we see. And in order to do that, we need to look for clues. So one way to look for clues is to look for the presence of inflammation. And we have these little point of care tests where we dab the ocular surface and we look for a pink stripe. And if you see the pink stripe, then you can say, hey, there's inflammation. But when it's faint pink, it's only a little inflammation. And I don't get off the couch for a little inflammation. But when it's fuchsia, that's when I start looking and saying, maybe inflammation is contributing to the dry eye symptoms and signs. Now, another way to look for inflammation is with these beautiful images I get from my confocal microscope. And there are these morphological findings called activated dendritic cells. And these are cells that we think are immune origin. And when they are activated, they grow in length. And they generally hang out here, touching the nerves or close to the nerves. And you can see here's an example of someone who does not have activated dendritic cells. This is another marker of inflammation and actually could be a biomarker of having a systemic disease associated with dry eye symptoms and signs. So I find this very helpful. I also look for blood tests that look that there could be an autoimmune component to dry eye. And I look for both established markers of Sjogren's syndrome, things like SSA and SSB, but then we have some newer markers that are not part of the definition, but that suggest to me that there could be an autoimmune component to dry eye. But beyond thinking just about inflammation, I also need to think about other contributors. Like, how can I detect if there are nerve abnormalities? And this is, again, something that I'm really focusing on. And when I say nerve abnormalities, you can have an abnormality in the first order neuron. That's the part of the peripheral nervous system. But you can also have an abnormality in the central nervous system. And the interesting thing is, if you have firing, inappropriate firing of neurons anywhere along the corneal pain pathway, you may feel like your eyes are dry, even if the abnormality is up here in your brain, right? So anywhere in this system can lead a patient to coming in and saying, my eyes are so dry. So what are my clues? Well, right now my clues are a lot of them borrowed from, in general, the field of neuropathic pain. I look for things like allodynia and hyperalgesia. Now, those are fancy terms for saying something that hurts me a lot, hurts someone with something that hurts me a little, hurts someone with neuropathic pain a lot. So if someone comes in and says, I'm so sensitive to wind, I'm thinking, sounds like hyperalgesia. Allodynia means that something that shouldn't hurt at all hurts someone with neuropathic pain. And so the clue I'm looking for is someone who comes in and tells me that room light kills them. Now, the other thing I do is I look for specific comorbidities, because what I have found is that pain travels together. So if someone comes in with a diagnosis of fibromyalgia or migraine, these are causes of chronic pain elsewhere, I start thinking, maybe the nerves are abnormal in the corneal pain pathway as well. And what I'm looking for in terms of signs is I'm looking for patients whose symptoms outweigh what I see on my exam. I use dental floss to look at corneal sensitivity, and what I'm looking for is abnormal sensitivity. And then I have this thing called the anesthetic challenge. Now, you may remember that we use anesthetic prior to checking pressure. And this is a way of using something that we already have in our clinic and repurposing it for another test. The other thing I do is I lightly touch around the eye. And if people have pain with light touch, that's also a sign of neuronal abnormalities. And we call that cutaneous allodynia. Again, that's a fancy word for pain when you lightly touch around the eye. So I use my Prepare Cane challenge to try to figure out if I think someone has a nerve abnormality, where is my nerve abnormality? And so if the nerve abnormality is in the peripheral nervous system, then the pain should go away when I put in a drop of anesthetic. But if it's in the central nervous system, or it's in the peripheral nervous system, but not in the corneal pain pathway, then the pain persists after Prepare Cane. And this is a very useful test for us to figure out, or to help us figure out, the location of nerve dysfunction. So the question is, why do I do this? Well, another important facet of dry eye is that we have to align treatment with the contributors. So if I see inflammation, I need to treat inflammation. And we do have a number of anti-inflammatories available to us. And there are lots of new meds that are being investigated in current trials. But we also need to recognize that there are other aspects of disease beyond inflammation, and we need to treat them when needed. So there are companies looking at treating nerve function or lacrimal output. And so we don't know which of these is going to be best for which dry eye subtype, but it's exciting that we have some investigators and companies looking at other aspects beyond inflammation. Now, one of my focuses has been on treating nerve dysfunction. And what I do is I go to my pain specialist and to my neurologists, and I say, hey, what are you using that may be helpful for my patients? And so for my pain specialists, I use oral neuromodulators, drugs that have been out there for a long time to treat patients with neuropathic pain outside the eye. And I repurpose these for the use of neuropathic pain within the eye. And then for my neurologist, I borrow adjuvant therapies like trigeminal nerve stimulation, like botulinum toxin, approved for migraine. But the reason I'm so glad you're listening today is because really we need more. We need better diagnostics. We need to subcategorize dry eye so we can individualize treatment. We need to diagnose disease earlier. We need more treatment options, looking at different aspects of disease. And I think kind of a window to the future will be disease prevention. And so I just gave you a lot of information. I want to show you how I apply this in my everyday life. So the question is, why does my patient have dry eye? And so I like to always start with the case. This is one of my patients. He's 66, and he comes in and he says his eyes burn and he's so sensitive to light, and he's unhappy about this. And so the first thing I want to ask is, who is he? And it turns out he has had multiple surgeries, both corneal surgeries and cataract surgery and retinal surgery. And when I see this number of surgeries, I get a little nervous. But fortunately, he has an excellent optometrist who put him in scleral lenses, and his vision is decent. But the problem is he's still having pain. He's very unhappy. So I really want to understand, what is my patient complaining about? And so he tells me he has constant aching with exacerbations, like a vice grip is on his eye. And I have these standardized questionnaires that I have my patients fill out while they're in the waiting room to help me figure out exactly the intensity and the quality of their pain. And the bottom line is he has what we consider severe ocular surface pain. And so I also want to know who my patients are beyond their eyes. So do they have immune diseases, pain elsewhere? Do they have devices that may exacerbate dry eye, like a CPAP machine? And it turns out he does have migraine and depression, and he's being treated for these conditions, but his other comorbid conditions are pretty well treated. And so I put on my detective hat, and my exam starts when I walk into the room. And so the first thing I'm looking at is his overall anatomy. Does he have any eyelid abnormalities? Does he blink? Are his eyelids in good position? What does his periocular skin look like? And you know, it's not bad. Here I am actually pooling up on his eyes, so it looks distorted, but his anatomy was mostly normal. Then I look for inflammation, and I do it with one of my point of care tests. And you see here that there's a little pink, but again, I don't get off the couch for a little pink. And then I have a battery of tests that I look at the meibomian glands. Remember, those are the ones that make the oil component that seals in the water component of tears. And turns out his meibomian glands look pretty good. Now, my magic weapon in the clinic is this thing called a fluorescein strip, because it dyes the tears a green color, and then I'm really able to visualize them. So I'm able to look at tear stability. I'm able to look at staining. That was the disruption I showed you in the other picture. And I'm able to look at tear production with my little Schirmer strips. And overall, these are very healthy metrics. So he really does not have a water problem. And when I look at the rest of his eye, he doesn't have a lot of injection. He doesn't have inflammation in his eye. He doesn't have pigment in his eye. And overall, he just doesn't look that bad. Now, my nerve metrics do show me that his nerves are abnormal. He has reduced sensation, and he has pain that improves with anesthetic, but doesn't totally go away. So probably both a component of peripheral and central abnormalities. My confocal scan doesn't show a lot of activated dendritic cells, but it does show that his nerve density is slightly decreased, but not terrible. So overall, he doesn't look as bad as I thought he was going to, but I need to figure out, why does he have so much pain? And so again, it's really important to remember that lots of different things can cause pain. So one source of pain are nociceptive causes, not making enough water, abnormal oil, and you have nerves that are normally responding to these abnormalities in your environment. But the second cause is neuropathic causes. The nerves themselves are abnormal. And in his case, I'm worried that he has a neuropathic component to his pain. And the reason I think that is because his corneal sensation is abnormal. He has persistent pain with anesthesia. He has low nerve density on confocal scan, and he also had pain to light touch around the eye. And overall, he had a disconnect between what he was complaining about and how he looked. And so what do I do when I diagnose a patient and I think they have a component of neuropathic pain? Well, I first still treat all nociceptive sources of pain, but then I move on. And the thing is there are lots of different ways to treat nociceptive sources of pain, and years of excellent research has gone in to developing these strategies. But also really important to realize, when your first plan doesn't work, it's important to move on and consider nerve dysfunction. And the reason this is needed is because I see many patients who were diagnosed with tear dysfunction, and it was only after a few eye care providers that nerve dysfunction was considered. And by the time they see me, they're frustrated, they're anxious, and they're saying, why couldn't have this been caught or thought of earlier? And so what do I do with these patients? Well, the first thing I do is provide education. There are no magic fixes for chronic ocular pain with a neuropathic contribution. Things happen slowly, and years of therapy are often needed, the goal being good enough. And when it comes to treatment, multimodal therapy is often required. And it's really important to realize that with the drugs we have now, the pain effect, the improvements in pain, it's not instantaneous. I describe it as a slow linear improvement, sometimes a sigmoidal curve. Patients need to know this going in so they have realistic expectations. They need to know that it's going to take time. Now, in this case, because the pain got a little better with my drop of anesthetic, I'm trying to target his corneal nerves. And historically, I use autologous serum tear or blood products for this indication. And maybe you've heard that lots of people use these products for various injuries. For example, Tiger Woods injected such products when he hurt his tendon, but we actually also use it for patients with corneal nerve abnormalities. We also have a new eye drop, recombinant nerve growth factor, but there's really no data on how it would be used in peripheral neuropathic pain, because it's currently approved for the other subtype of nerve abnormalities, which was the neurotrophic subtype. But again, this is why research is so important, because there is a company that's now looking at a novel ocular pain eye drop. It's a TRPV1 antagonist, and it's for people who have pain after surgery that have at least a component of peripheral pain. And so fortunately, this is a randomized study. I don't know if he has an active drug or placebo, but he does have less pain, and that makes me and him very encouraged. So what is the punchline? The punchline is dry eye is a pain because it's a complex disease. You need to think about tears and nerves and need to treat specifically. And in order to do that, we need more research and research funds to achieve our holy grail, which is to deliver precision-based medicine in dry eye. So thank you so much for hanging out with me. And hopefully you realize that our goal is really to improve quality of life, and we need everyone's help to get there. So thank you very much.
Thank you, Anat. I know a lot of people have experienced dry eye symptoms, and recognizing the umbrella of dry eye conditions is really helpful. The impact on patients is vital, and I really appreciate you sharing the value of research and addressing all the areas of dry eye disease. With research in mind, I'd like to introduce our next speaker, Tannen Schmidt, who will share more about how research is addressing dry eye disease. Tannen is an associate professor in the Department of Biomedical Engineering at the University of Connecticut Health Center. His basic and translational research interests lie within biomechanics, biomaterials, biointerface science, and biochemistry and biophysics, all within the context of engineering health. This includes the ocular surface in health and disease. He was on the contact lens materials design and care subcommittee of the TIFOS International Workshop on contact lens discomfort, as well as the tear film subcommittee of the TIFOS Dry Eye Workshop too. Tannen?
Hello, everyone. Thank you for the opportunity to present at the Dry Eye Awareness Month Congressional Briefing. Today, I'll be discussing the question, is it possible to find a cure for dry eye disease without National Eye Institute funding? I've got my disclosures on my slide here related to Lupus Biopharma. So here's a brief outline of the presentation. I'll start with a brief introduction and background on the ocular surface in this molecule I study called proteoglycan-4 or lubricin. Then I'll move into discussing PRG4's lubricating ability at the ocular surface and on contact lenses, as well as its biotherapeutic potential in the context of dry eye disease. Next, I'll share work and studies related to recombinant human PRG4, specifically its expression and purification, and then success in a dry eye clinical trial. Finally, I'll end with the role and importance of National Eye Institute and the role it plays in supporting basic and mechanistic research. Being a biomedical engineer, I thought I would start by explaining what biointerface science and biotribology is, because it's particularly relevant here. A biointerface is an interface between a cell, tissue, or biomaterial with another tissue. We have many in our body at many different length scales. Biointerface science contributes to the understandings of interactions between biomolecules and surfaces and are central to science and bioengineering and crucial in research related to many fields. One of these fields is biotribology, which is the study of lubrication, friction, and wear of sliding biological surfaces. One such biointerface where the study of lubrication is relevant for dry eye is the ocular surface. Now, the ocular surface cells are subject to significant friction forces when they blink. When healthy, our tear film provides lubrication through a variety of components, including mucins, ions and proteins in water, and lipids. Now, the previous two speakers discussed in great length what dry eye disease is and the impact it has on people's lives. I'll just briefly restate that dry eye results in discomfort, visual disturbance, ocular surface damage, which may be due to increased friction or wear, and it affects millions of people worldwide, including contact lens wearers. Now, ocular surface lubrication may play a role in the initiation and or progression of dry eye disease. A specific example of this is how altered lubrication may be involved with dry eye is this tissue wear observed on lids called lid wiper epitheliopathy, which could be due to deficient lubrication or increased friction. Additionally, there is data that demonstrates of all the things that we can measure in the lab on contact lenses to try and understand contact lens discomfort, friction is one important variable that is associated with discomfort. It's for these reasons and others why there's been a focus on research and industry on the development of lubricating eye drops as potential treatments for dry eye disease. Now, this molecule I study in the lab and have been for the past 20 years is called PRG4 or lubricin. PRG4 is a mucin-like glycoprotein originally discovered in synovial fluid, that's the fluid in our knee joints, as a product of cells that line the surfaces. It was found at many other biointerfaces bodies. Basically, most places in the body where a tissue slides against another tissue. PRG4 is present on cartilage, tendons, spine, skin, heart, and others. Now, PRG4 is mucin-like because it has many short sugar chains capped with a negative charge within its central domain, and these have been shown to be critical for its various functions. Additionally, PRG4 demonstrates the mucin-like ability to dimerize or multimerize through bonds at the ends of the protein, which have also been shown to be important for function. Now, from a mechanical perspective, PRG4 functions as a boundary lubricant. That is, it reduces friction between surfaces in contact, much like Teflon in our pan when cooking an egg. It is also anti-adhesive. It is essentially Mother Nature's boundary lubricant at various biointerfaces in our body. Now, historically studied in the context of joint, it's now known that PRG4 is necessary for our knee joint health. Shown here is an image of cartilage on the left, and it's the cells at the surface of the cartilage that express PRG4. Lack of PRG4 expression in humans occurs in a genetic disorder called CACP, resulting in permanent flexions of the fingers, joint arthropathy, joint deformity, and even inflamed pericardium. These people suffer from joint deformity and ultimately early joint failure, in some cases, even adolescents requiring total arthroplasty. Cartilage deterioration and early joint failure is also observed in the PRG4 mice that lack expression, collectively indicating that PRG4 is critical for our joint health. Now, I won't speak more about PRG4 in the context of joints, as this is Dry Eye Awareness Month, except that there have been many, many studies demonstrating PRG4's ability through injections into our knee joint to prevent cartilage degeneration or arthritis progression, and this continues to be an active area of research. Now, with this years later, with my experience and expertise in PRG4, led to this wonderful discovery with other colleagues that PRG4 is actually present on the ocular surface and in tears, where it contributes to ocular surface health. So shown on the left is a human cornea and conjunctiva with the purple color showing staining of PRG4, and you can see it lines all of those surfaces. On the right, you see the PRG4 knockout mice, that is, it does not express PRG4, demonstrate increased staining at the ocular surface, suggesting that PRG4 deficiency may play a role in promoting corneal damage. So this discovery led to a whole new area of work evaluating friction forces, which I had expertise in the context of joints, and now was going to apply to ocular surfaces. So we took the test that we had developed for cartilage and modified it, taking the approach to use real human ocular tissues. This included various tissues and materials that would allow for different biointerfaces of interest, including a cornea eyelid, cornea contact lens material, or contact lens eyelid. So the cornea lens or eyelid lens was mounted onto a biomechanical testing machine such that it had a geometry to study boundary lubrication. Essentially, we could apply a force, rotate the other side, and measure torque, which would enable us to calculate a friction coefficient. So initial studies here focused on showing PRG4 provides boundary lubricating function at ocular surface biointerfaces. So shown here is a kinetic friction coefficient. And when these are lower, it means friction is lower or it's better lubricated. And you can see this was measured over a variety of sliding velocities. On the left, PRG4 has lower friction than saline and even lower than hyaluronan, which is common in over-the-counter eye drops. On the right is just showing that PRG4 lubricates in a dose-dependent manner, meaning the more PRG4 we had in solution, the greater reduction in friction we observed, going from 0 all the way up to 300 micrograms per mil. We then went on to evaluate friction at the cornea versus different biomaterials present in commercially available silicone hydrogel contact lenses that are common today. And again, in both cases, we won't go into details of these biomaterials, just to say that PRG4 reduced friction compared to saline on these common contact lens hydrogel materials. The next step with this is we decided to develop a new friction test employing a new mechanical testing machine where we mounted the actual eyelid edge on a holder and then a contact lens on this curved surface. This was motivated by this picture I showed earlier where we had the lid wiper epitheliopathy. So this eyelid could then linearly reciprocate over a curved surfaces where forces could be measured and then transposed into a normal and shear force, resulting in friction coefficient calculation. This proved useful to study commercially available contact lenses in a variety of contexts. Now, lastly and importantly, I want to focus on studies related to recombinant human PRG4. So full-length recombinant human PRG4 was successfully expressed by Lubris back in 2012 in collaboration with a protein expression company in Switzerland called Celexis. Now, this drew considerable attention in the news media at the time, as some other larger pharmaceutical companies had tried and failed to express full-length PRG4. So work in my lab went on to show that this full-length PRG4 had the appropriate higher order structure, size, and charge that one would expect for native PRG4. Now, I won't go into the details of the data shown, just to emphasize that based on these gels, we were able to demonstrate it had the appropriate structure and charge with those sugars. And over on the right, indicating size, that this molecule had a hydrodynamic diameter of approximately 170 nanometers, which is actually quite large for a protein of this mass. So structurally, it was similar to that of native PRG4, but importantly, functionally, it was as well. So we then went on to demonstrate that recombinant human PRG4 demonstrated similar boundary lubricating abilities at the ocular surface compared to that of native PRG4. So lastly, with this recombinant human PRG4 available and characterized in vitro, the next step was to assess its clinical utility in treating dry eye. This was done through a collaboration between an Italian pharmaceutical company and Lubris, and was published back in 2017. So the purpose of the study here was to assess the efficacy and safety of recombinant human lubricant as compared to a 0.18% sodium hyaluronate, or HA, eye drops in subjects with moderate dry eye disease. Now, I am not a clinician, but I can briefly go over the clinical design. So 40 patients were recruited and then split into two arms, one receiving the PRG4 at 150 micrograms per mil, the other receiving the commercially available HA eye drop. The first week, they were administered therapy 2 times a day. And then in the second week, they were free to administer therapy as often as they liked. And then finally, in the third week, they all crossed over to hyaluronan treatment. So the summary of the clinical trial are shown here. Primary endpoints were met, such that PRG4 supplementation achieved a greater than 72% reduction in symptoms from baseline. These included foreign body sensation, burning and stinging, pain, sticky feeling, blurred vision, and photophobia. Additionally, PRG4 showed significant improvement compared to the hyaluronan in terms of fluorescein staining, tear film breakup time, sandy frequency of symptoms, where you can see a drop of approximately 40 points, eyelid and conjunctival arrhythmia, and installations. Now, the limitations of the study was 1, the small sample size, and 2, it being single site. However, the conclusions that could be drawn was that PRG4 was found to demonstrate significant improvement in both signs and symptoms of dry eye disease compared to HA control. Now, with this exciting findings and my move here to the University of Connecticut at a similar time, I continued basic research into trying to understand the mechanism of action of PRG4 and really started to learn and appreciate just how important National Eye Institute funding is to make this happen. So motivated by the clinical trial and the fact that 1/3 of the patients in that trial had Sjogren's syndrome with dry eye, we then went on to demonstrate that PRG4, when exposed to cathepsin S, which is an enzyme upregulated in tears of Sjogren's syndrome and has been suggested to be a biomarker, completely lost its ocular surface lubricating ability. So what's shown here on the right is that when you expose PRG4 to cathepsin S, which is up in Sjogren's syndrome tears, it completely degraded the protein. Then when we took this degraded protein and assessed its friction reducing ability on our mechanical testing device, you can see that PRG4 plus cathepsin S was similar to that of saline and significantly higher friction than the PRG4 alone. Now, given the elevated activity of cathepsin S in Sjogren's and the role intact PRG4 plays in health and lubrication, degradation of PRG4 could be a potential mechanism for diminished ocular surface lubrication in Sjogren's syndrome. Now, to further follow up on this, we wanted to test if levels of PRG4 in tears were different or deficient in Sjogren's tears. So tear washes were collected from healthy and Sjogren's patients, which were then analyzed for PRG4 by a custom bead-based assay we had developed using PRG4 as a standard. The results demonstrated that indeed PRG4 levels in Sjogren's syndrome tear wash were significantly decreased compared to normal, indicating that PRG4 could be a biomarker for Sjogren's or provide rationale why PRG4 supplementation in Sjogren's syndrome patients improved signs and symptoms of dry eye. So we've continued to study PRG4 to try and understand its mechanism of action. And we did this in the context of experimental dry eye with collaborators, as well as in context beyond just lubrication. So we published a paper last year with colleagues showing that first, PRG4 expression in the ocular surface in mouse experimentally induced dry eye was significantly diminished, as you can see here at the surface. We also showed that human corneal epithelial cells could actually internalize the PRG4, which could be a potential mechanism by which the PRG4 acts on the cells. Another important finding of this initial study is that 1, PRG4 has anti-inflammatory properties, and that it's not just another lubricant. These results demonstrated PRG4 inhibits inflammation-induced expression of molecules that can attract immune cells. So shown here is the white bars is the absence of PRG4, and these molecules stimulated by various inflammatory stimulants. When treated with PRG4 in the black bars, you can see a reduction in a variety of circumstances. Another interesting finding was that PRG4 binds to and inhibits the enzymatic activity of MMP9, which is a clinical biomarker for dry eye disease and inflammation, in which dry eye patients often have elevated levels. While these are interesting and exciting results, there's still much to be understood. So coming back to the role and importance of National Eye Institute funding for dry eye research, I'll start with the mission. The mission states to eliminate vision loss and improve quality of life through vision research. Indeed, dry eye disease significantly affects the quality of life of millions of people, and there remains a need for effective treatments. Now, basic and translational academic research requires funding for people, whether it's researchers, students, fellows, or support staff, reagents and supplies, core services and equipment, and of course, the ability to publish and share results with the community. So to finish and come back to the question of this talk, is it possible to find a cure for dry eye disease without NEI funding? In my opinion, the answer is no, which is why continued and increased funding for dry eye research from NEI is so very, very important, and is something that I will continue to strive for. Thank you very much.
Thank you, Tannen. That insight into the research being done to address dry eye disease is really compelling and shows the value and impact that research is having and can have to address this disease. I want to thank everyone for attending today. We're running a little tight on time, and so I want to thank all of our speakers for their help in bringing greater awareness to dry eye disease, the lifestyle factors and implications for dry eye disease, and the research that is working to manage and address dry eye disease. The National Institutes of Health through the National Eye Institute continues to support advancements in the research of dry eye. With the prevalence of dry eye disease likely to continue to grow, research into lifestyle implications, interventions, and therapies are essential as we look to improve, manage, and cure dry eye disease in the future. The investments we make today in research pay dividends in the future, and the National Eye Institute is central to funding advancements in vision science. I also want to again thank our supporters who helped make this event possible, including Research to Prevent Blindness, TIFOS, and ARVO, and to Genentech and Novartis for providing event support. Thank you all again, and I hope you have a great afternoon.